In the last video, we surveyed pathways used to produce ATP. Oxidative phosphorylation, one of these pathways, provides almost all of the energy for aerobic cells. For example, we obtain 95% of the energy we consume from oxidative phosphorylation. During oxidative phosphorylation, many of the electrons feeding the electron transport chain are released in the citric acid cycle bound to universal electron donors such as NAD and FAD. The citric acid cycle is fed by acetyl coenzyme A. In this video, you learn how acetyl coenzyme A is produced. If you remember, glycolysis takes place in the cytoplasm and leads to the formation of two molecules of pyruvate. Under aerobic condition, pyruvate is converted into acetyl coenzyme A inside the mitochondrial matrix. So acetyl coenzyme A is a key metabolic intermediate linking glycolysis and the citric acid cycle. A carrier protein transports pyruvate into the mitochondria. And then acetyl coenzyme A is formed by oxidative decarboxylation. So why is the formation of acetyl coenzyme A called an oxidative decarboxylation? Well, it's kind of self-evident. Pyruvate is decarboxylated and oxidized, hence oxidative decarboxylation. So during the oxidative part of the process, two carbons from the pyruvate will form an acetyl group that is attached to coenzyme A. And in the decarboxylation part, the third carbon is removed to form carbon dioxide. Oxidative decarboxylation is catalyzed by a multi-enzyme complex called the pyruvate dehydrogenase complex, which is made of three types of enzymes, E1, E2, and E3. In bacteria, the PDH complex contains a catalytic core formed by 8 trimers, each made of 3 E2 subunits. Each one of these 24 E2 subunits is divided into 3 domains: a flexible lipoamide domain, a transacetylase domain, which is responsible for the catalytic activity of the enzyme, and a small domain allowing interactions between E2 and E3. The PDH complex contains also 24 E1 subunits organized into 6 tetramers and 12 E3 subunits organized into 6 dimers. The number of E1, E2, and E3 subunits varies from species to species. For example, bovine PDH contains 60 copies of the E2 subunits.
I assume that the PDH complex is huge, but can you give us a sense of its size?
Well, in mammals, the PDH complex is relatively large. It's larger than a ribosome. Of course, if you don't know the size of a ribosome, it does not tell you much. The diameter of an eukaryotic ribosome, which is considered as a very large protein complex, is about 250 to 300 angstrom, while the diameter of the PDH complex is about 500 angstrom. To function, the PDH complex requires 5 additional coenzymes. Thiamine phosphate, or TPP, is a coenzyme covalently attached to E1. We have covalently attached to E2 a coenzyme called lipoate, or lipoic acid. And transiently bound to E2, we have coenzyme A. FAD is covalently attached to E3. And NAD+ is transiently bound to E3. So our 5 coenzymes are TPP, coenzyme A, lipoate, FAD, and NAD+. The oxidative decarboxylation can be divided in 4 steps: decarboxylation, oxidation, transfer, regeneration. These steps are catalyzed by different PDH subunits, and the coenzymes that I just mentioned a minute ago contributes to the catalysis. Before we explain in detail the steps of catalysis, let's consider a little bit more the conversion of pyruvate to acetyl coenzyme A. This is a very exergonic reaction which is irreversible. Carbon atoms from glucose during glycolysis are incorporated into pyruvate and then will be incorporated into acetyl coenzyme A, or into the acetyl group bound to coenzyme A. These carbons that derive from glucose will have two fates. Either they will be fully oxidized in the citric acid cycle, or they will be incorporated into fatty acids and then lipids.
So acetyl coenzyme A is used to form fatty acids. Does the irreversible conversion of pyruvate into acetyl coenzyme A mean that living organisms can't make their own sugar from fats?
In animal, it's true. And in fact, this is a great observation. Since oxidative decarboxylation is irreversible, sugars can eventually be transformed into fatty acids, but fatty acid cannot be converted back into sugar. Plants and some microorganisms have a unique pathway called the glyoxylate cycle that leads to the conversion of fats into carbohydrates. Unfortunately, animals don't have this cycle. So now let's describe in more detail the 4 steps of oxidative decarboxylation, starting with the decarboxylation. It is a process that is catalyzed by the E1 subunit and implicates the coenzyme TPP. The carbon from the cyclic structure of TPP, located between the nitrogen and the sulfur atom, is very acidic and can be deprotonated and react with pyruvate. We have the formation of a transient covalent intermediate that is then decarboxylated and reprotonated. So the outcome of this reaction is the release of carbon dioxide and the covalent attachment of an acetyl group to TPP, forming hydroxyethyl TPP. The second step is the oxidation of the hydroxyethyl TPP. This reaction is once again catalyzed by E1 with the contribution of the lipoamide arm, which is bound to E2. It is the transfer of an acetyl group from TPP to the lipoamide arm. During this process, a disulfide bond on the lipoamide arm is broken. A thioester between lipoamide and the acetyl group is formed, and the other sulfur form a reduced thiol group. The outcome of this reaction is the regeneration of TPP in its original state and the formation of an acetyl lipoamide. Before we move to the third step, let's spend a minute on the formation of the thioester bond. You've probably heard about ester groups that contain an oxygen atom, and they are called oxygen esters. In our case, the ester group contains a sulfur atom instead of an oxygen. That is why it's called a thioester. Thioesters have a very large standard free energy of hydrolysis. And one could say that the formation of a thioester bond in the acetyl lipoamide activates the acetyl group. Why is a thioester bond in acetyl lipoamide more favorable than an oxygen ester bond? Just look at the free energy diagram. You can see that the free energy of a thioester is higher than the free energy of an oxygen ester. Why is that? Well, in an oxygen ester, electrons are delocalized around the 2 carbon-oxygen bonds, leading to a resonance stabilization. The presence of the sulfur in the thioester prevents this resonance stabilization. The thioester is less stable, and in consequence, the delta G prime naught for the hydrolysis of a thioester is more negative than that for an oxygen ester hydrolysis. So the presence of this thioester bond will facilitate the transfer of the acetyl group from the lipoamide group to coenzyme A. And that is the third reaction in oxidative decarboxylation. This reaction is catalyzed by E2, and the transfer is made possible because the acetyl lipoamide has a high energy potential due to the thioester bond. So one thioester bond is broken during the process, but one new thioester bond is formed between the transferred acetyl group and coenzyme A. The outcome of this reaction is the formation of one molecule of acetyl coenzyme A that has a very high energy potential. The delta G prime naught for hydrolysis is minus 31.4 kilojoule per mole. And we also form one fully reduced dihydrolipoamide. So after step 3, the disulfide bond on lipoamide was reduced and has to be regenerated. Otherwise, the PDH complex could not complete another catalytic cycle. The regeneration is catalyzed by E3. E3 carries FAD that will accept 2 electrons and 2 protons released when the disulfide bond on lipoamide is regenerated. So FADH2 is formed. But once again, we need to convert FADH2 back to FAD for the next catalytic cycle. And that is done by the reduction of NAD into NADH plus a proton. Once the reduced NADH is released in the mitochondrial matrix, it can be used during oxidative phosphorylation. So now our reaction is complete, and the PDH complex is back to its original state. So let's go through the steps of acetyl coenzyme A synthesis again, this time looking how different parts of the complex work together to channel the substrates through the reactions. So in the first step, you remember that pyruvate binds to the active site of E1. Carbon dioxide is released, and the acetyl group is attached to TPP. The active site of E1 is connected to the surface of E1 by a 20 angstrom long hydrophobic channel. So our hydroxyethyl TPP, the product of the first reaction, is at the bottom of this channel. During the second step, the flexible arm of lipoamide that is bound to E2 will penetrate into the channel in E1. It will bring the reactive part of the lipoamide arm very close to hydroxyethyl TPP. And E1 will catalyze the transfer of the acetyl group to lipoamide. Upon transfer, the lipoamide arm swings out of the E1 active site and enters the catalytic core formed by the E2 subunits. Now we have the third step. You remember that E2 catalyzes the transfer of the acetyl group from lipoamide to coenzyme A, which is bound to the active site of E2. The acetyl coenzyme A is released, and a fully reduced lipoamide arm is formed. In the last step, the reduced dihydrolipoamide swings from E2 to E3, where the disulfide bond is regenerated, FADH2 is formed and then reoxidized again by transferring its electrons and protons to NAD+, that was bound to E3. So once NADH is released, the PDH complex is ready for the next catalytic cycle. The clustering of enzymes and coenzymes in a single complex increases the catalytic efficiency by limiting the diffusion of reaction intermediates. This is an example of substrate channeling. So now, how is the formation of acetyl coenzyme A regulated? The synthesis of acetyl coenzyme A is the gateway to oxidative phosphorylation and fatty acid synthesis. So we can make some predictions. the PDH activity depends on the energy state of the cell and will be inhibited at high energy state when the concentration of ATP is high, and activated at low energy state when the concentration of ATP is low. And because we are clever, these predictions happen to be true. There are two levels of regulations of the PDH complex. The first type of regulation is allosteric regulation. At high energy state, you have a high concentration of the immediate product of the reaction, acetyl coenzyme A and NADH, as well as the ultimate reaction products, ATP and fatty acids. These four molecules will act as allosteric inhibitors of PDH. For example, acetyl coenzyme A acts on E2, NADH acts on E3. At low energy state, when ATP is needed, the substrates of the reaction, coenzyme A and NAD+, are at a high concentration. AMP will be also at a high concentration. And these three molecules will act as allosteric activators of the PDH complex. The second regulation is by reversible phosphorylation. So let's consider what happened in muscle. In muscle, you have a kinase and a phosphatase. The kinase, PDK, phosphorylates the PDH complex on E1. This phosphorylation leads to the inactivation of PDH. Then the phosphatase, PDP, will dephosphorylate and thus activate the PDH complex. At high energy state, reaction products, acetyl coenzyme A, NADH, and ATP, activates the kinase and thus inhibit PDH. At a low energy state, for example, when there is muscle contraction, the reaction substrates, pyruvate and ADP, will inhibit the kinase PDK, preventing further inhibition of the PDH complex. And calcium, which is released during muscle contraction, will activate the phosphatase, therefore activate PDH. In other tissues, hormonal controls of PDH activity is observed. For example, in liver and adipose tissue, we know that these two tissues funnel glucose to acetyl coenzyme A and ultimately to fatty acid. In the liver, the binding of epinephrine to its receptors leads to an increase of the intracellular concentration of calcium that activate the phosphatase, PDP, and ultimately activate PDH. In the liver and adipose tissues, a high blood glucose leads to production of insulin, which stimulates the phosphatase, activates the pyruvate dehydrogenase complex, leading to the conversion of the excess glucose into fatty acids. In the next video, an animation will lead you through the 8 steps of the citric acid cycle and will help you to follow the fate of the carbon atoms from an acetyl group entering the cycle through not 1, but 3 turns of the cycle.